Our today's topic is C4 and CAM pathways of carbon dioxide fixation. C4 and CAM pathways are alternate pathways of C3 cycle to fix carbon dioxide. In C4 cycle, the first formed stable compound is oxaloacetic acid. Oxaloacetic acid is a 4-carbon compound. That's why it is called C4 cycle. C4 pathway is also called as Hatch and Slack pathway, as Hatch and Slack both worked out the pathway in 1966. C4 pathway is commonly seen in many grasses, sugarcane, maize, sorghum, amaranthus, etc. In CAM pathway, first formed product is malic acid, and plants exhibiting CAM cycle is called CAM plants. For example, Bryophyllum, Crassula, Opuntia, orchids, etc. C4 plants. First, C4 plants show a different type of leaf anatomy called crane's anatomy. Here the chloroplasts are dimorphic in nature, means two types of chloroplasts are present in the leaves of C4 plants, right? Here vascular bundle, you can see here in this diagram, vascular bundles are surrounded by bundle sheath cells. These are bundle sheath cells, dark green, these, right? These are large parenchymatous cells. And these bundle sheath cells have chloroplast. And chloroplast of bundle sheath cells are larger, lack grana, but contain some starch grains, right? And the chloroplast in these mesophyll cells are smaller, have grana. So this type of anatomy is called crane's anatomy. C4 cycle involves two carboxylation reaction, right? One taking place in chloroplast of mesophyll cells and other in chloroplast of bundle sheath cells. So C4 cycle has 4 steps: carboxylation, breakdown, splitting, and phosphorylation. First, carboxylation. Carboxylation means it is a chemical reaction in which carboxylic acid is formed by treating a substrate with carbon dioxide. In simple words, addition of carbon dioxide, right? So here, this carboxylation step is means it takes place in chloroplast of mesophyll cells, right? So here phosphoenol pyruvate is there, which is a 3-carbon compound, right? When it picks up carbon dioxide in the presence of water and phosphoenol pyruvate carboxylase, means PEP carboxylase enzyme, it changes into oxaloacetate, a 4-carbon compound, plus phosphoric group, H3PO4. So this is carboxylation. Next step is breakdown. Now, oxaloacetic acetate, which is formed in this reaction, carboxylation, then breaks down into two compounds, aspartate and maltate. Both are a 4-carbon compound in the presence of two enzymes, transaminase and maltate dehydrogenase, right? Transaminase and maltate dehydrogenase, both enzymes are required here, right? Then these two compounds which are formed diffuse from the mesophyll cell to bundle sheath cells. Third step is splitting. In bundle sheath cells, this maltate and aspartate split enzymatically to yield free carbon dioxide, means decarboxylation is there, means removal of carbon dioxide, plus pyruvate, which is 3-carbon compound. So carbon dioxide produced here in this reaction is used in Calvin cycle in sheath cells for the production of sugars, right? And last step is phosphorylation. Phosphorylation means attachment of a phosphate group to a molecule. So here, pyruvate molecule, it is transferred to chloroplast of mesophyll cell then, and here it is phosphorylated, means addition of phosphate group in the presence of ATP and pyruvate phosphokinase enzyme. And a product which is formed is phosphoenol pyruvate plus AMP and pyrophosphate, right? So this is C4 pathway. Here, addition of carbon dioxide, right, means carboxylation, and its product is oxaloacetic acid, 4 carbon compound, right? Then splitting of this oxaloacetate means breakdown of oxaloacetate into 2 compound, aspartate and maltate, both of 4 carbon compound, right? Third step is splitting. When this maltate is transported to the chloroplast of bundle sheath cells, here decarboxylation occurs, right? Means removal of carbon dioxide. And this carbon dioxide then enters to the Calvin cycle, and pyruvic acid is formed. This pyruvic acid then transported to the chloroplast of mesophyll cells, and this pyruvic acid, and last phosphoenol pyruvic acid is formed, right? Pyruvate molecule then transported to the chloroplast of mesophyll cells. Here it is phosphorylated, right? Phosphate group is added, and then phosphoenol pyruvate is formed, plus AMP plus pyrophosphate, right? So this is C4 pathway. Now, crassulacean acid metabolism means CAM pathway. CAM is a cyclic reaction occurring in dark phase of photosynthesis in the plants of Crassulaceae family. Its first product is malic acid, and it occurs in the mesophyll cells, right? Plant exhibiting CAM cycle are called CAM plants. These are some examples like Bryophyllum, Crassula, Sedium, Cactus, like Opuntia, and some orchids, right? CAM involves 2 steps, acidification and deacidification. Now, CAM plants we know are succulent, and they grow under extremely xeric conditions. So here leaves are fleshy or succulent, right? So mesophyll cells have larger number of chloroplasts, and the vascular bundles here are not surrounded by well-defined bundle sheath cells. Okay, so in these plants, stomata remain open during night and closed during daytime. So these plants are adapted to photosynthesis and survival under adverse or xeric conditions. Okay, so in first step, that is acidification, here stored carbohydrates in darkness, stored carbohydrates like phosphoenol pyruvate are converted into phosphoenol pyruvic acid by the process of glycolysis. So when the stomata of CAM plants open in dark, they allow this free diffusion of carbon dioxide, right, from atmosphere. This carbon dioxide, when picks up by phosphoenol pyruvate in the presence of water and PEP carboxylase enzyme, oxaloacetic acid is formed, plus H3PO4, phosphoric group. Oxaloacetic acid, which is produced, then reduced to malic acid in the presence of NADPH2 and malic dehydrogenase enzyme, right? Deacidification then. During the daytime, when stomata are closed, this malic acid is decarboxylated, means removal of carbon dioxide in the presence of some malic enzyme, right? So malic acid plus NADP, product is pyruvic acid, NADPH2 plus carbon dioxide. So this is CAM pathway. Here first, phosphanol pyruvate is there, right? When carbon dioxide enters in the presence of PEP carboxylase, oxaloacetic acid is formed. You can see here in this. So then maltate, and maltate as a malic acid then stored in the vacuoles. This malic acid, which is produced in the dark, is stored in these vacuoles, right? So malic acid then increases the acidity of the tissues. That's why this step is called acidification. And during deacidification, malic acid is removed and it decreases the acidity of the cells. So here decarboxylation is there, carbon dioxide is removed and it can be used in the Calvin cycle for the production of sugar, right? So if we compare C4, C3 and CAM plants, C4 plants are more efficient in photosynthesis than C3 plants. Why? Because enzyme phosphanol pyruvate carboxylase of C4 cycle is found to have more affinity for carbon dioxide than ribosco enzyme, ribulose diphosphate carboxylase oxidase of C3 cycle in fixing molecular carbon dioxide, right? So CAM plants also are not as efficient as C4 plants in photosynthesis, right? So this is all about the CAM pathway. Thank you.